Hi, my name is Susan Messino. I'm a joint appointment in neuroscience and psychology at Trinity College, and I'm delighted to be part of the NIH Virtual Symposium on Metabolism-based therapies for epilepsy. And I'm going to give you kind of a high-level view of the efficacy of metabolism-based interventions in various animal models and their impact on human studies. I have no conflicts to declare. So metabolism-based therapies is really a very rare opportunity where the efficacy was first observed at the bedside with the ketogenic diet in 1921. And there's been decades of work trying to understand how it works at the bench and in research laboratories. One of the issues with developing these models for metabolism-based therapies is that there's such a mixed bag of variables, diet compositions, outcomes. So there's a lot of, it's a big data space to try and unpack and understand. And one of the really challenging things is that even though this is the rare opportunity where we know something is effective clinically, there have been many instances where metabolism-based therapies have been ineffective in our animal models. And that's true in some in vivo models and also in some in vitro models. So that's something that needs to be taken into consideration, obviously, if we're trying to understand how metabolic therapies work. So I wanted to start with some basic questions about why you would pick a particular animal model and how you would pick the model. And of course, you would need to justify the use of any animal model as being a good model for your particular mechanism that you're looking at or type of epileptic condition. And I want to emphasize at the outset the additional focus on rigor and reproducibility when the NIH has published these guidelines, principles and guidelines for reporting preclinical research. So this is an attempt to really try and standardize and increase the rigor of preclinical research so that we can have the best possible translation between animal models and clinical applications. So when you're thinking about an animal model, of course, the animal kingdom is so incredibly vast, and animal models have different strengths, different evolved adaptations, and different clinical relevance. About 95% of our animal research is done in rodents, and one of the things that's been a challenge with rodents is some different characteristics of their metabolism compared to humans. And I think what we're trying to do now as a field is put together pieces from different animal models to try and get the best information possible. In terms of the reliability of an animal model, I wanted to try and sort of look at both sides of the coin from a paper from Geyer and Marcu in 2000. So one of the important things to take into consideration is what is the consistency and the stability with which the variables of interest are observed. So with epilepsy, obviously we're looking at seizure frequency, seizure duration, seizure progression, and what is the ability to manipulate the independent variable with a high degree of accuracy and measure the dependent variable objectively. Ideally, you want small within subject variability and small between subject variability so that you can reproduce the phenomenon under similar conditions and reproduce the effect of manipulations. So this is one thing that we're looking at with kind of rigor and reproducibility. But I also wanted to introduce another aspect to think about, and that is that epilepsy is inherently very variable. And one thing to take into consideration is that while a reliable and reproducible system is essential, it should be emphasized that the variability could even be part of the system. And I want to emphasize this statement that the study of the variability could lead to a better understanding of the phenomenon. And in some cases, variability may not be necessarily an error. It's an inherent property of what we are studying. And I think epilepsy is really an example of where this is something to take into account, in that we know that metabolic therapies don't work for every patient and don't work for every type of epilepsy. So that's an important consideration. In terms of in vivo models that have been used to study metabolic therapies, they range all the way from flies, fish, rats, mice, and there's a broad array of ways these models have been used that we don't have time to discuss in the context of this overview. And I also wanted to point out that there's also a number of in vitro models, so ranging from single cell systems, acute slices, chronic organotypic slices, and even in vitro models of dictyostelium or slime mold. So really spanning the whole range of the animal kingdom. And what we found when we just tried to do sort of an overview of the results, even just with in vitro applications of ketones themselves in slice models, is that we found that there were a range of results where some labs found positive results on seizure-like activity, and some labs found no effect of applying ketones. So even in this reduced system, within rodent models, there are a wide range of different findings. In terms of in vivo models of ketogenic diet or metabolic therapies, there are a wide range of models as well. So this is just a very brief kind of survey of electroshock-induced models. You can see a range of papers. There's drug-induced seizure models, a range of papers for that. And this is just a very kind of cursory survey. Genetically modified animal models, typically mouse models, and a variety of kind of seizure etiologies within this category, and also opportunities for applying ketones in animal models of a range of rats, mice, direct application, gavage, etc. So this is a big data space with a lot of variables. And the power, of course, of rodent research is the range of genetic manipulations, phenotypes, and the powerful kind of work that's already been done. So one of the things that I think is coming to the fore is that two things that may be most helpful in translating animal models to human epilepsy. Number one is genetic models that are either directly representative or replicate conditions in epilepsy. So the top paper is just an example of where the ketogenic diet improved outcomes in a model of SUDEP, sudden unexpected death in epilepsy. So that's a mouse genetic model. In the lower paper is an example where the ketogenic diet was able to prevent epileptogenesis and disease progression in adult mice and rats in a progressive model of epilepsy that could mimic the progression of epilepsy in humans. So some of the kind of overarching questions to consider are, we know that a lot of our decisions are driven by constraints. So research is guided and driven by technology and models, what's available, what's possible. And if we want to try and accurately translate metabolic therapies to human experience, what seems to be coming to the fore, if we're talking about rodent models, is that genetic and/or chronic progressive models can really help us to identify mechanisms that are evolving as part of the disease process. I think a question that we should continue to ask is how can we develop important new models and who can help? So researchers like us can help through the peer review process. Research institutions can help with new models via support of appropriate facilities and infrastructure. And also funding agencies can help to develop new models by encouraging more exploratory and high-risk approaches to develop these models that could really help us to help inform clinical translation. I wanted to make a point about an additional in vivo model that I think has a lot of promise, and that is using community-based dogs. Dogs do have a high incidence of epilepsy, and they share a lot of similar lifestyle factors as we do. And I think it's an opportunity that's been underutilized in terms of development of metabolic therapies that has a lot of potential. So in summary, just for this overview, in general, metabolic therapies and ketogenic diet is really a rare opportunity where we know it works at the bedside, and we're trying to use the bench to find out how we can really take advantage of having this metabolic intervention help with more people, either by optimizing a metabolic approach or developing an analogous therapy. We know that these metabolic therapies mobilize numerous mechanisms. So that's sort of a complication, but also a real strength. And within that data space, the animal models are many, in vitro and in vivo, with many variables. And we need to make sure that we attend to rigor in our design, preclinical criteria, and what would really be ideal is if we can find key biomarkers to help to translate. We know that advancing the field is going to require rigor within and integration across models. So this is an important thing to keep in mind. And in future work, we're going to want to align these variables and cross-validate, and particularly look for biomarkers that can help us to really leverage the research interest and the public interest in this bench to bedside and bedside to bench translation of metabolic therapies to help patients with epilepsy. So with that, I'd like to thank you for your attention, and I look forward to your questions.